Hi class. Now I am starting a chapter on environmental biotechnology. Environmental biotechnology, as you know, has come into limelight uh, since last many years now, a few decades, I believe, from early 2000. And climate change, both scientists, uh, people in public offices and non NGOs, non-governmental organizations, they're all coming together to highlight the significance of climate change um, on our lives. And this awareness is increasing, and it has further come into mainstream discussion because of recent pandemic, which is supposed to be, which is being linked to climate changes, and I would say lifestyle change and many other things. So, environment is essential aspect that reflects the changes in the climate. It could be washing away of seashores, changing of temperature, which temperature which is not compatible to other living organisms, and our humans are being considered to be the main players who are responsible for bringing these lack of natural resources or dwindling natural resources. The awareness is now being made at international stage, at international forum, to pay attention to the climate change. Otherwise, there will be a point that there will not be any returns, and it will be further, it will become bad and bad. So what could, as a biotechnologist, or what could a scientist do to prevent environmental, let's say, pollution in simple terms, how to deal with it? So now there are many biotech companies that are coming up that are taking responsibility. They are playing a frontier role to understand and develop products that could um, remove those elements from the environment which could be harmful to humans. And one of those companies is named as Biotechnica, that are working to treat the pollutions. So there are two directions in which environmental biotechnologists are working. Number one, uh, is to develop develop instruments that would enhance detection, right? For example, lead in our water is not being marked as a good sign for health, because it could induce liver toxicity, it could induce many other toxicity in our body. So develop instruments that would detect low quantity of lead or any other metal in our in air or in water that we consume. The second emphasis of environmental biotechnologists is to develop uh, tools such as in the form of lower organisms, lower organisms that can degrade, or that can consume uh, pollutants, right? They could convert something that is toxic into a non-toxic byproduct. So there could be many more, but these are major. To develop sensitive instrument. So these sensitive instrument could be biosensors, right? That can detect small speck of lead in air or in water. And then, for example, to develop, let's say, bacteria, E. coli bacteria, that would degrade toxic pollutant from the environment or from the water. And and these bacteria are non-harmful. They're not harmful. So they can treat the sewage system, the water system. And the second could be, algae that can, if you remember the BP spill that happened in the coast near New Orleans, that had a huge impact on the living beings of in that area. So scientists developed recombinant algae that would degrade those crude oil in that area, and without causing any pollution, because it is impossible to clean those things. And cleaning using machines or some other tools can create another kind of pollutions. So to have something organic. So these are the two areas on which environmental biotechnologists are focusing. And one of the companies I shared is Biotechnica. So advantages of bioremediation. There are a number of cost-effective advantages to bioremediation, which can be employed in areas that are inaccessible to without excavations. So it's not possible for a scientist, for a worker to reach, but yes, if they employ bacteria or some plant, then it could be. For example, hydrocarbon spills, as I mentioned towards BP, the sea level became so thick that it was not possible for a human to reach there. So they needed some organism, they need some tools to reach in that thick area of the water. Okay. So these are few of the bacteria or organisms that has been developed to take care of compounds that could be harmful. So remember, if you remember the shape, you don't need to know the chemical structure, but I'm just saying, if you remember the shape of the nucleotides. So nucleotides are of two types, purine and pyrimidines. And if you look at their shapes, so they are something, so purines are double ring structure, right? Something like this. Talking of purines, which are adenine and guanine, right? And there are purines that are single ring structure molecule. Now, if you look at a lot of pollutants, these pollutants resembles these nitrogenous bases, purines or pyrimidine. So it is hypothesized that these pollutants can easily, affect our DNA. So much so that these these pollutants can mimic the DNA nucleotides, and these pollutants can insert themselves into our DNA. And when these pollutants, when they insert themselves into our DNA, if this is the normal DNA, then if these pollutants, they enter the DNA, then the DNA would become kink. Kink means if this is the helix, then it will be really sudden, sudden kink, sudden eruption because of these bases. And because of that, the DNA would not be normal. So if there is a transcription, translation, or replication, these DNA would give rise to mutant form of DNA and the mutant protein, and then many diseases, including cancers. So that's the immediate effect of these pollutants. So bacteria, first bacteria can be made. It hardly, once a bacteria, recombinant bacteria is made, it hardly needs special or expensive reagent to maintain, and they can be recycled. They can keep on growing in the lab, and they can organically degrade these pollutants, right? So we were talking about bioremediation. And so some of the bacteria, such as Acinetobacter or Pseudomonas, they can degrade or digest trichlorophenoxide, acetic acid, and stuff like that. These are hydrocarbons, the carbons that can affect lungs, our DNA, and that can induce mutation, which could cause long-term defect. So I mentioned to you earlier a word, biosensors. So what are biosensors? Biosensors are devices that can detect compounds, inorganic, organic compounds, even in the tiniest amount. So there are various technologies that are coming together. For example, recently they have developed nanotechnology, which could be more sensitive. They can screen more number of samples. So biosensors is essentially, it emerged from the marrying of two disciplines, biology and physics, or instrumentation, and it can help to detect. So let's say if there is an area in New York City which let's say by chance there is a bad water supply, and we don't know what is there in the water, and let's say there is a mixing of flood water or something, or sewage water, and we need to know the purity. So these biosensors can detect at a very, very low amount the prevalence of these pollutants. So now they are not only used for pollution, they can also be used for diagnostics, right? If they want to, let's say, detect a COVID RNA into our plasma or in our body, these sensors can be extremely efficient. So not only for detecting uh, pollution, but let's say presence or absence of sugar, presence or absence of a bacteria in our body system, these biosensors could be extremely efficient as a diagnostic tool. So many technologies, many disciplines of science, physics, computational, biology, they come together to develop an instrument that has direct effect on human health and diseases. So this was also a very small chapter, but an important one for future, because climate change is directly affecting us if it is occurring. Yes, it is a topic of discussion, debate, but as a student of biology, as a student of biotechnology, we should think about how to develop tools, how to design experiments, or to do research in what area, because this is going to be a very important upcoming area of research in days to come. Okay, so thank you.